Hello everyone, this is Dr. Vishal Trivedi and this is our second interaction sessions. So if you have any questions, you can ask me. I will be happy to answer your queries. आवाज आ रहा है? So we have first questions from the Kalpana, and what she is asking, I think she is asking about question where have I asked for how many peptide fragments you will get if you are going to digest with a particular amino acid sequence. So as you know that the trypsin is always cutting the peptide fragments wherever it contains the lysine or the arginine. So if you see in that particular sequence, we have actually three places where either the lysine or the arginine is present. So if you have three cuttings of a peptide fragments, you are actually going to generate the 4 fragment, and that is what the correct answer is. I do not have the sequence right now, but if you want to verify the answers, what you can do also is you can actually take that particular peptide sequence and put it into a web server called as peptide cutter. So if you go to the peptidecutter.org and if you paste the sequence, and then the peptide cutter is actually giving you the flexibility of choosing what are the different enzyme you want to choose, whether you want to see the sensitivity of this particular peptide fragments with a particular protease or not. So if you do that and only choose the trypsin, what you will going to get is the number of fragments and the places where it is going to cut. And if you do so, it will actually clarify your confusion as well. So if you have any more questions, you can ask me. I will be happy to answer. And I hope the Kalpana might have been satisfied, or I think you can right away itself you can try out the peptide cutter and see that the answer is matching the answer what we have given you on the against our assignment as well. Uh, Now we have another question from Mr. Biplab, and what he is asking is assignment marks allotted is not clear. Can you explain it? So, in what way you are looking? You want me to explain you the allotment in terms of the allotment as per the per assignments, or it is like, because as you can see in the initial when the course started, the number of questions what we were giving were more, because more and more when we started the course, it was more of a descriptive in nature, but later on it was become more in analytical in nature, and that's why the number of questions got reduced. Initially we were asking around 20 to 25 questions we were giving you. That's why the initial assignments were the initial assignments were of 20 to 25 marks, but then later on when it becomes more analytical, then the number of questions got reduced, because that time we would like to check how much you have understood the concepts and all that. So that's why the number of questions got reduced and the marking also got reduced. But overall it is actually very relative, because if you have attempted all the assignments, you will get the marks accordingly. So if that will help you to understand, if that is clarified your stuff, I am sure I will be fine, but otherwise you can ask me again if I have not addressed your query. And we have one more question from Kalpana as well, and what she is asking is what is happening in dye affinity chromatography? Is our desired product get colored? The first thing what you have to understood is that dye affinity chromatography means that where a particular dye is going to be coupled to the matrix, and this particular dye, when it is coupled to the matrix, has a exclusive affinity for a class of molecule. For example, we have given you an example of Sebagreen 3GA, and Sebagreen blue is, which is actually when it is bound to the matrix, it is making a blue dextran, blue saffrose, sorry. So it is giving you the blue saffrose, and that blue saffrose has a very high affinity for NAD plus containing dehydrogenases. In fact, it can bind NAD plus containing other enzyme as well. So it has affinity for that NAD plus cofactor acquiring enzymes. So it can be used for all those class of molecules. So as I think, and your second question is that whether the product is going to be colored. So that is not true, because when you bind the proteins, it will actually go and bind to the dye, and when you are going to elute the proteins, only the protein is going to be eluted, not the dye. Dye will still remain to the matrix, because if that will happen, then your column is going to be destroyed. So in a dye affinity chromatography, the dye will be still remain bound to the column, and it will never come out. So your column is not going, your product is not going to be colored at the end. Okay, so we have another question related, or I think it's a kind of a query whether I will do that. So the Mohit is asking, will you be provide question material for exam including paper pattern, etc. So for that purpose only we have the module 12, because in the module 12 we are actually going to take up the revision of the whole course. So the module 12 actually has the 2 lectures. In the first lecture I have covered whatever I have taught you in this course from module 1 to module 6, and in the lecture 2 I have covered all the material what I have taken. In terms of the very briefly, I have touched all the concepts, and I at the end of the 12th module, when you will see the assignments, that assignment will be actually the same pattern what you are going to see in the exam. So in this exam, what we are going to have is anyway will be the multiple question type of exam or MCQs, which it is actually the objective what you are going to see, and the exam is going to be of 100 marks, like you're going to have the 100 questions. So the questions are and so it will be like objective type, where you have to choose the right answer, and it should be so when you will see the assignment of the module 12, that actually will give you a little idea about what kind of exam pattern you are going to see in this course actually. The other question is so there is a query from one student. She is saying that the course should have been more focused on the genetic engineering. So that is what we have tried in the beginning, because genetic engineering, what you will learn, we have covered that in detail, and then later on we have tried to see whether you can actually also should understand the downstream processing as well, and how the once you got the product be formed within the cell, how you can actually purify and utilize that product for biotechnology application. So that's why I thought if I add that, it will actually going to make the story complete, instead of genetic engineering and saying that okay, your gene is cloned and so on. Because once I take up the downstream part also, it is actually going to give you more it will actually going to cover up all the troubleshooting what you can do, even if once you clone the protein as well. Then as I have attempted for CAD exam in September. So I am not very clear. Can you clarify, Mohit, what you are asking? I don't know what you are talking about. And as far as the Mr. Shah Viplav is also concerned, what you are saying is, as per assignments 15 questions are set, but in the score chart it shows variation of marks. So total marks per assignment, what does that mean? Means you are saying that even if the total number of questions are 15, but you are getting 13 or 12 or the separate numbers, like can you clarify that, what you are really want to ask me? Okay, so for Mohit, I have a simple answer. Simple answer is there will be no sections in this exam, in the final exam when you are going to attempt it for the certificate. It is going to be a one section only, and that section is going to contain the 100 questions, and all these 100 questions you have to attempt. So there will be no section that you can choose from section A, section B and section C, because that is more applicable when I have the population of different streams attempting the different types of subjects. Here the subject is only the genetic engineering and you know, so it's going to be only one section, or actually the single paper which you have to answer. I hope it is now clear to you. Should we'll okay, so for Mala, I think if you are going to go through with the notes, that is good enough for clearing the doubts, or that is good enough for clearing the basic informations. But as far as the exam is concerned, the exam is definitely going to cover up even beyond the scope of this, beyond the syllabus. For example, as I said, biotechnology applications I have covered as much as possible, but there are many biotechnology applications which are very popular, and that is also you should know. So, many of the questions which I have put in the exam paper are more of like a general knowledge type of questions rather than the course type, for example. So, those things are pretty much but other than that, most of the questions are from the course material. Most of the questions are from the lecture notes what I am providing along with the video lectures. Don't worry about the notes, because we are in a process where we are actually trying to make the notes and trying to provide as much as we are possible, because so far the transcripts are also not available for this particular course. So, I understand that it is difficult for the students to cope up with the amount of video lectures what we have and the other text. So, they really need reading material for that. So, that's why it is we are trying that you should get the reading material as well in terms of the notes actually. So, we are preparing the notes for the course, and we are posting as much as it is available with us. I know that still you don't have the notes for the initial 1 or 2 modules, and that also we are preparing and we are going to post in due course. But later on, I think you are going to get the notes for everything by the end of this course. So, and your exam is anyway in November, so you are going to have sufficient time to go through with all these notes, and you can also see the videos very quickly as well, and then that will actually going to definitely help you to prepare for the exams which are going to be in November. And as far as the book is concerned, what you can consult is what I am teaching is actually from the many books, but for basic genetic engineering you can consult any standard molecular biology book. For example, gene, or you can consult the molecular cell biology or those kind of textbooks. And for biochemistry, you can easily consult the lineages and other kind of textbooks, and that should be good enough to cover up the course. It's for 1 hour, right? Uh, so for Mala, the question what you are asking is, I think, perfectly correct, and I think this is the question which I think we have floated in this assignment itself, or I think the last assignment. So we are not going to discuss much about the answer and other things, but as far as the question is concerned, this is completely correct. Um Okay, there is no tip for remembering the things. There is no way that you can remember the things without memorizing, and on the other hand, there is no need to actually memorize things, because once you understand the process, you understand the how it happens. By if you go through with multiple times, actually it will be, you don't have to remember that. If you understand it properly, you will be able to work out even without knowing the intermediates and other things. For example, in Krebs cycle and other places. Uh, so I think I won't suggest that you should remember or memorize the things, because when you memorize the things, they don't last for very long, but when you understand the things, that understanding remains with you forever. So what I will suggest is, you should understand the relevance of the Krebs cycles, you should understand each and every reactions, how it is happening and all that, but don't try to memorize it, because nobody is going to test your memory at the end of this course anyway. What we are going to test is something else. What we are going to test is your analytical abilities. What you are going to test is how much you have understood and followed during the course. If you could have seen the assignment as well, most of the assignments, sometime I ask the assignment where the memorization is important, and that is only because I want you to go through with the video lectures, I want you to go through with the content as well. So that's why I just ask those type of questions. But as far as the exam is concerned, we are not going to ask the questions to test your memory, because some memory, memorizing or something is very easy task. Understanding a concept is a very, very difficult thing to do, and that is what our objective is. We would like to explain you, we would like to make you understand. Even if you know that, okay, there is a ligation reactions after you get the sticky ends from your insert as well as the vector. Suppose you don't you will not be able to remember the name, whether it is T4 DNA ligase or T7 DNA ligase. That is a very, very minor mistake. Instead of that, you don't know even that there is a ligation reactions required and how the ligation reaction works, what are the cofactors are required in the ligation reactions. So that is just a simple example that you should focus more on the process rather than on the the participants which are participating in that particular reaction, because the process, understanding a process is very, very important. Now the okay, so Kalpana is asking why we cannot use or why cannot we call the Comasi billion blue or Comasi dye as a tracking dye. So if you remember, we are not using that Comasi dye while we were actually running the samples, okay, because the Comasi is going to bind the proteins, okay, and the Comasi will not going to tell you how much you have run the gel, and so you don't want to make a complex of the Comasi dye with the protein while it is running, because that actually is going to affect the overall charge and overall many properties of that particular protein, because you know that the friction forces are going to be modulated if you stain the dye, they stain the proteins while it is running as well. So that's why we don't call the Comasi as a tracking dye, because Comasi compared to that, you use the bromophenol blue dye, which actually does not run, does not run along with the proteins. It does not make a complex. The other point is, if you add a dye which actually makes the complex with the protein, it will actually going to tell you that okay, this is the running of the protein, but how much gel has run, that actually it is not going to tell you. So there are two different things. How much the solvent has gone, that actually you have to know, because otherwise the protein will run out from this SDS page. So for that you need a dye which should not react with the protein, and it should have of a very low molecular weight, so that it should run really fast, and it should be the first molecule which you can see in the gel. And that's why we are using the the bromophenol blue, because it is blue in color, so it is visible, and on the other hand it does not bind the protein, so it is not going to make a complex, so that its binding or its migration is also going to be retarded. And on the other hand, it is going to tell you how much the solvent has run into this particular gel, so that you know that the protein is anyway not going to run beyond this. So once it reaches to the end of the plate, you can just stop the electrophoresis and remove the gel from your sample. I hope you could have understood, and I could have been able to clarify the things why the Comasi is not being called as a tracking dye, because it makes a complex with the protein, and then you are actually tracking the migration of the protein instead of migration of the solvent front. If you want to monitor the migration of solvent front, you are looking for a dye which is very small in molecular weight, and that should not make a complex with the protein. Yeah, you can use the xylene xylitol as well for tracking, but mostly xylene xylitol people don't use for SDS page, that they use for always for the agarose. And but yeah, you can be able to use xylene xylitol as well for tracking. In gel filtration chromatography, the smallest molecule get inside the pore of the beads. Okay. So when you are flowing more and more liquid, okay, then there is another round of distribution. Then again you know that initially when you are loading a sample, the samples are competing for each other, and that's how the smallest molecule is filling into the inside of the pore, and the bigger molecules are sitting on top of them. Okay. But when there is no supply of molecule, because your sample is going to be get exhausted in due course when it is loaded already the column, then the sample molecule, then your solvent molecule is going to flow through these beads, and the solvent molecule is nothing but the water actually, or even the molecule which are smaller than the smallest protein, and that is how they are actually trying to compete with these molecules. So, that is how these molecules will start. So, there will be a distribution of this molecule, whether the water molecule will sit into the pore or the small molecule like the smallest protein, and that is how the smallest molecule will start coming out from the pores. And once they come out from the pores, they have to run all the way from top to bottom, so that they will be come into the detector, and that is how they are going to show you a peak, and that is why the small molecule comes late and the large molecule comes first in the gel filtration. So, time is running short. In case you have any more questions, you should ask me, because you should utilize this time to ask your queries, to understand each and every concept. What I could see that many of the people are simply more worried about the exams and assignments and all those things, but what they are not interested, or what they are not be much worried about the subject as well. So, it is better to you ask me questions related to the subjects rather than so it is possible. Can you clarify? I think Shekhar, can you clarify what is that mean? Is it possible that smallest molecule may not be completely recovered from the column and are lost during the process? It is not possible, but it is only possible in those conditions when the smallest molecule is getting stuck into the column, and or sometime when you have a very small quantity of the smallest molecule, because when you load 100 microliter or 200 microliter protein the mixture onto a gel filtration column of 25 ml, there will be a 100 time dilution of these fractions. So, suppose you loaded only 10 micrograms, so ultimately it is going to be reached to a nanogram level. In those cases, it could be possible that the smallest molecule may not be get lost because it is not coming out, but it may be get lost because your detector may not be able to detect the fractions. So, you may be collecting the fractions, but when it was coming in a very low quantity, the detector was not showing any reading, and that is why you might have ignored to collect that fraction. But other than that, if you have loaded a sample and the sample does not have any affinity for the column, there is no way that it can get lost from the column. It will definitely going to come out. It is a different issue whether the detector will be able to give you the readable reading, so that you will be able to collect the fractions or not. I think you can do the both. What I will say is that you can revise as per the you can revise using the notes what I have provided. You can revise the notes what you have prepared. In fact, when the person makes the notes on themselves, those notes are always been better than the notes what is been provided by the teacher, because when the teacher provides the note, it is mostly on the by the understanding of the teacher itself, but when you prepare the notes yourself, that may be better than the teacher's notes. So, it is your choice, you can choose my notes or you can choose your own notes. The ultimate target is that you should understand the concepts and you could be able to clear the exams. If you have any more questions, please ask very quickly. The session is getting over or going to be over very soon. I think affinity chromatography is based on the mutual and exclusive interactions, or I will say the interacting residues present on the cognate pairs. For example, if you have a ligand, it may have a specific three-dimensional conformation, and that specific three-dimensional conformation of the ligand is going to fit into a particular receptor. And why that? That is because that receptor has the complementary conformations. On the other hand, the receptor has the several sites which are actually making the interactions very stable. So, because of that, this particular ligand and pair can be used for affinity chromatography. You cannot use any two ligand and any receptor, because one of the important thing is that the ligand receptor interactions or the ligand receptors what you are going to choose for affinity chromatography should have very high affinity, very strong affinity, which should not get even get affected by simply by putting the NaCl or some other kind of the electrostatic interactions and so on. It should have large sufficient affinity, so that it should not get affected very soon when you change the buffer conditions or so on. For example, antigen and antibody. So, antigen antibody interaction is so strong because the antibody has a complementary three-dimensional conformation what the antigen is having, and on the other hand the antibody is also having the specific pairs which are actually binding the antigens and which are helping in making this interaction more and more strong. And because of that, if you use these particular pairs, they will be able to help you to purify the complexes. Sir, can you clarify what should be score in assignment for eligibility for exam? Okay, so there is no hard and fast score. I think it's already given on the announcements. What you can go through with the announcement, and that will tell you that what is the score. But I think what we are going to do is, if you attempt all the 12 assignments, we are going to take the top 9, which wherever you have done the good, we will take only those scores, and then only we are going to conclude whether you have done sufficient enough to get the certificate or not. As far as I remember, I think in the assignment it is 30% what you have to score. So, suppose out of the 12 assignments, first of all we are only going to check the 9 assignments, and then out of 9 assignments you only have to get the 30% marks. And then in the score in the exam also, I think you have a percentage which you are going which you get, and if you do also if you get this much enough marks, I think you will get the certificate at the end of this course. What you are doing actually, Surabhi, what you are doing now, even suppose you are, if you are you a BTech students or MTech students or you a PhD student, can you clarify that? if you're not, I think, see, after completing this course, there are many avenues one can explore. For example, those who are doing the BTech or MTech courses actually can pursue the PhD and higher courses. With these courses, so with that, and on the other hand, since if you might have seen the videos, I have taken a lot of, I have incorporated many experiments in this course. So, if you go through with those experiments and if you see how the technique is being performed inside the lab also, then actually it will give you a very first side experience that how to perform the technique as well. So, once you know the theory, you know how to perform the techniques, that actually makes you very valuable assets whenever suppose you do this course and you join any biopharma company, or suppose Dr. Reddy's lab or some other groups, and suppose they were trying to use the recombinant DNA technology to produce some protein for vaccinations or some other kind of applications, then you will be very useful for them, because then you can be able to you can be able to pick up the things much faster and much quicker compared to those who only have the theoretical knowledge or those only have a practical knowledge, because you are going to have both the knowledge by doing this course. So, best of my knowledge, even if you get the more than 90 marks or 90%, I do not know if there is a additional opportunity or any special opportunity for those students. You may get you may get a different type of certificates, but the purpose of NPTEL is to provide you the best education and best experience and best thing possible through an online mode. Apart from that, I do not know if there will be additional advantage of scoring more than 90 in the NPTEL courses. So, I think you are asking about screening of gene by immunological method. So, screening of immunological method is simply as it like you are doing the Western blotting. Okay, as you are using the antibodies to detect the different types of proteins. Similarly, in this case, what you are going to do is you are going to first make the cDNA library. As you can know that the immunological method is not applicable for genomic library, simply because of that the reason that the genomic library may contains the constructs which may not be get expressed. So, for that reason only the immunological method is more appropriate to screen the cDNA library, because those are the constructs which are going to express the proteins. So, what you are going to do, you are going to first make a master plate. From the master plate you are going to make a replica plate, and then for the replica plate you are going to incubate that replica plate in a lysis buffer, and once you incubate them in a lysis buffer, the bacteria or yeast or whatever the organism you are using for making the cDNA library are going to get lysed, and the protein which is present on inside that particular organism is going to get immobilized onto the membrane. Once that process is over, then you are going to use the primary antibody to detect the protein which is going to be produced by the gene of your interest. Once you do the primary, you can do the secondary antibody which is coupled to the HRP or alkaline phosphatase, and then you will apply the substrate. Once you apply the substrate, you are going to get the signal from the clones which are actually going to contain the protein of your interest. This means the gene of your interest, and then you can go to those clones You can do further dilutions, or you can use the clones as such in case it is a single clone, and you can be able to get the desired gene using the immunological screening method. So, if you all having any questions, we have another 1 minute left only for this session to conclude. So, if you have any questions, very quickly you should ask. And as far as since Surabhi has now said that she is pursuing the BSc from Allahabad University, what I will suggest, what I will suggest, okay. So, for Surabhi, I have only thing what I can tell you is that after doing this course, probably what you can do is you can either appear for either you can pursue the MSc, or you can go to the higher studies, and that actually in this course whatever you are going to learn, it is actually going to help you in getting the higher courses. For example, if you complete the MSc, then this course, if you complete this course, it is actually covering a large chunk of the GATE exam. So, if you see that syllabus what I have covered almost 40 to 50% of the syllabus what is being covered in the GATE exam itself. So, this is actually going to help you in terms of preparing for the GATE exam as well, because the only thing what is left which is I have not covered is the immunology portion and the little bit the biochemical engineering. Rest all related to recombinant DNA technology or the chromatography or analytical techniques and all that we have covered in large in detail. So, that is how this course is going to help you. And as far as your question about the air channel is formed, so the air channels and air pores, okay, these are the same thing. Air pores are actually the smaller portion where you are going to have 1 or 2 air bubbles, whereas the channel is something where you are going to have a larger surface area of the column which does not have the water, which means it actually is a very big air bubble actually, which actually been formed. And I have another query from the Mr. Biplav Saha. So, what he is saying is that in the assignment 8, he secured 87 out of 15 questions. So, actually what is happening is that every assignment, irrespective of whether it is 20 questions or 30 questions or 100 questions, every assignment is been given the 100 marks. So, you are getting 87 out of 100 actually, because at the end we have to equalize. So, suppose some people, if that will not be the case, those who have attempted my initial assignments where I have given 20-25 questions, they will get good marks compared to the people who might not have attempted those assignments. So, for that purpose only each assignment is being considered to be of 100 marks, irrespective of whether you give 15 questions or 18 questions or 22 questions or 25 questions, because it distributes accordingly. So, you are going to get, suppose if you attempt all the 15 questions and all the 15 questions you are going to get the correct, then you are going to get the 100 marks. That does not mean anything, because 100 marks means 100. So, for example, if even if it is a 20 questions, you are going to get the 100 marks. Even if you are going to get 15 questions, you are going to get the 100 marks. So, it is actually the percentage corrected answer from your side. So, in your case, the 87% you have made a correction or corrected attempt out of the 15 questions. I hope it is clarified you. I am not sure about IIT JAM because I have not seen the syllabus of IIT JAM, but if you take the biotechnology, maybe it is possible that it might have covered some sections. So, I do not know about that. As far as I know that it has covered a pretty large chunk of courses or the questions what people ask in the GATE exams. So, I think if there is no more question from the student side, we would like to conclude our session here, and I am very thankful to you all, that to all those who have attended the sessions and asked me all the questions, and that actually helped the okay. So, I am not very sure about what you are asking, Shekhar. So, what you are saying is the partition chromatography is always superior to adsorption chromatography. It depends actually the type of molecule what you are purifying, and it also depends on the many parameters. So, partition chromatography is always superior, that I am not sure about that. And I also have no idea what for Mr. Saha also, that says after this course can I go for postdoctoral course. So, definitely you can pursue that. This course is anyway of a higher level course actually. So, this course is prepared in considering the BTech and MTech both courses. So, you are anyway pursuing a course which is of postdoctoral level actually. So, once you clear this course, you can definitely be able to go to the postdoctoral courses. So, with this I think I would like to conclude our live sessions, and if you have any more and further queries, feel free to post the questions in a discussion forum, and I will be happy to help you and happy to answer all the queries. Thank you.